Welcome to the second K.U. lecture. First, I would like to invite Professor Zhang Anchi to introduce the history and spirit of this lecture.
首先，于院士有跟我联系，他很希望能够在现场接待我的讲座。因为讲座有几个比较特别的精神啊，我想这个的缘起大约是6年前，我记得是2015年，我去 Oxford 接受他们的一个学生的 club， 叫做 Cortex Club， 是他们的神经科学的 graduate student 的 club， 那邀请去演讲。我看到这个的时候，通常本来是在想，已经很忙了，不要去。但是他就跟我讲了一件事情，就感动了我。他说这是他们全校的 student 投票出来的然后投票他最想听的国际上的任何一个人的演讲。我一看，另外3个都拿诺贝尔奖，所以赶快就自掏腰包，不管怎么样都一定要去那我说，哇，人家这么好的大学，给一个这样子的 honor， 那 honor 是无价的。刚好在那个时候，伍焜玉拿了这个总统奖大家知道总统奖是台湾这个科学最高的荣誉那总统奖的精神就是 promote 基础研究。我想很多老师可能慢慢已经没有感受到基础研究的重要，对不对？大家都说要创业，要赚钱，但是总统奖的 spirit 还是回到科学的最基本的面，优先的去认同最顶尖的基础研究。那伍院士，我们和他这个国卫院院长退下来之后，一直在清大的生命科学院合并那他拿了总统奖以后，他就很希望能够把这样子的一个精神能够落实，所以他当初说，刚好我在这个负责这个生科院的时候，他就说，那希望把他总统奖一半来帮助生科院 promote 基础科学研究。可是其实蛮困难的基础科学研究是蛮一般的，对不对？广泛的那我们总不能再给一个总统奖对不对？所以当时候就刚好这2个事情激发了我们这样子一个想法，是不是台湾回到最基本的从一个 bottom up 的，假如我们能够模拟那个 spirit， 找一群邀请一群最基础的清华的这个后起之秀也许他们都是下一个 generation 的大师，一起来问问他们最想要多有 interaction 的中生代的 leader 在全台湾是谁那当然这边有很多学生，顺便广告一下，也许大家还没有很清楚的。其实这个委员会也欢迎大家 nominate 把你们最想听的送给。那现在委员会的 chair 是林玉俊所以明年度你们觉得台湾你们最想听的演讲是谁？把这个提名给这个 committee 去做的讨论。这样做是也希望从不同的城市能够真的找到最好的这样子的讲座的研究人员一起来 promote 基础研究。那去年他们讨论出来是林颂兰那林颂兰 MD PhD 那一年刚好又发表一个 paper 在 SAY， 所以慢慢的就有一个这个 role model 在这个地方。那今年经由 free discussion 那一致的推崇我们蔡金吾教授他也非常的一连串的杰出的工作。 这个委员会还有一个很特别的地方哈，就是说像李彦俊做得非常好，大家一直希望他当主席。那但是从 Day One 哈，我们就自己内部规定，深圳教授以后就要离开这个委员会，让新的杰出的年轻的研究人员进来，所以李彦俊就很努力的想要赶快。那其实我们不是给奖啊，我们是邀请，很主动的邀请台湾最杰出的科学家一起来 promote 台湾下一个世代的最杰出的科学研究。所以我待会蔡金吾的介绍会有委员会来介绍，那以上我来代替分玉来介绍说为什么要有伍焜玉这个学术讲座的一个精神。好，谢谢大家。
感谢江安世院士的介绍。那接下来我要跟大家介绍一下这一届的讲者蔡金吾博士。This year, the committee selected and invited Professor Cai Jingwu to come give us a lecture. Professor Cai is a professor at National陽明交通 University. His group studies neurodevelopment and stem cell using the state-of-the-art technologies. And while their research is still basic science, their research can be applied to developing new therapeutic strategies for neurodevelopment disorders, neurodegenerative diseases, movement disorders, and brain tumors. Professor Cai received many awards in the past, including but not limited to Outstanding Research Award from Ministry of Science and Technology, Epilepsy Research Articles of the Year, and Epilepsy Research Award from Taiwan Epilepsy Society. 癫痫 Epilepsy 是癫痫。李天德 Award for Medical and Pharmacological Technologies, Academia Sinica Research Award for Junior Research Investigators, and YZ Xu Science Paper Award. And he currently serves as a Dean of Research and Development at National陽明交通 University 研发长。In addition to all these accomplishments I just described, I'd like to share with you what I found when the committee visited him earlier this year. First, his office was super clean and organized. And second, he was super prepared for the interview. He even left a space for us to put on our camera. And while preparation doesn't guarantee success, I often see this kind of attitude among successful people and would like to share this with you. So now, I would like to invite the Dean of College of Life Science to give Professor Cai this award. Although we don't really call it an award, but I don't know how to describe it. 接下来我们请生科院院长高瑞和院长来颁奖。感谢院长颁奖。那接下来就要邀请蔡金吾博士介绍他的研究，然后请大家要仔细听讲。结束之后，前几名问问题的同学会有 7 的礼券，这是 bonus， 平常没有的。Now it's time for Professor Cai to tell us about his research. Let's welcome him, and also for students who don't understand Mandarin. Please note that the first few students asking questions after the talk will receive gift card from 7-Eleven. Okay, let's welcome Professor Cai.
So I've never been so nervous giving a talk. First, I would like to really appreciate Professor Kenneth Wu for this great lectureship, because this is, to me, one of the greatest honors that I received, and especially, I think this is very, very special because usually we come up here and get a plaque or something like that, and then it's done. But today, I have some work to do. Okay, and entire day actually. Not only this morning, I need to give a scientific talk, but in the afternoon, meet with all young faculty and also the student members. But actually, I'm more excited about meeting with students and young faculties this afternoon because this is for me like I've been doing this for hundreds of times, and so this is like routine to me. But meeting with new students, like new faculty and students, I think that would really make a difference because I have a lot of things to tell you about myself and about my story. And I think behind research, there are a lot of things that I think the students may not know, when you attend to, scientific lectures. So, this morning I would like to focus on science, but in the afternoon, if you have time, please come join us because I'm going to tell you about my personal stories and up and downs of my research career. Okay, so I hope that will benefit, and maybe shed some light on your really dark life in your master or PhD research. Okay. Since I'm sure that there are a lot of students and also even faculty who do not understand or do not actually participate in this brain development research. So I will be really, really brief and not going into all the details. So it will be a very general, okay? So if you have questions, like in-depth questions, then we can discuss later. So today I'm going to talk about using various genetic and optical approaches to investigate mechanisms and genes that are involved in brain development and also disorders, okay? Because my background is actual physics. I graduated from NTU with a physics and zoology bachelor degree. So to me, interdisciplinary research is like gene inside me, because I think if you start early, you can switch really, really quickly, from physics to biology. I think there are really, really different disciplines, because when I was in college, physics, I need to do calculations and all that. But stepping into the biology class, I have to memorize everything. Okay, that's very, very different mindset. So I didn't get a very good score in the beginning, but now I sort of can master it because you keep going and going and you understand how to understand biology. Okay, so before I start, I will give you a really brief animation of how the brain develops and also how the nervous system developed in the mammalian system. So this is a human embryo, and in the 1 month of pregnancy, there are not many cells, and also the membrane is very tiny. It's even smaller than a $5 coin. And the cell number is about 10,000, and at that time there are only, most of them are progenitor cells. And then the frontal part of this neural tube would actually enlarge and develop into this cerebral cortex. And this cerebral cortex is actually very, very complex, as Professor Jiang would appreciate, because we have so many cells, so many neurons, 80-something billion neurons, okay? And Dr. Jiang, how many neurons do a Drosophila have? No. What? 100,000, so this is 80 billion, okay? So it's very complex. in our brain. And we are interested in how this brain develops. But there are many other tools that we can study. So how do we investigate this? So from my perspective, I started to think maybe I can start from disease. So in humans, there are diseases that are associated with neurodevelopment. For example, in this patient, the brain is smaller. This is called microcephaly, or smaller brain. This brain is called lissencephaly, the smooth brain. On the surface, you see there is a lack of convolutions, these wrinkles. And it turns out there are many genes associated with these diseases, and we have genes, we can study them. So this is really quite exciting. So one of the genes that I was studying in my PhD was called LIS1, the lissencephaly gene. And these patients are really suffering from a lot of symptoms. For example, epilepsy, developmental delay, learning disabilities, and all that. And so you guys are really lucky, okay? Because our brain are more or less normal, so that we don't have these symptoms. But if we want to understand how the brain develops and also how genes defects would cause these diseases, it's still very difficult to study human. So this is where basic research comes from. So if we want to understand it, well, we can use rodent models, one of the good models mimicking our brain. And when we dissect out the brain, and we can see how the brain, how the cells are located. For example, in this developing brain section, you can see there is a developing cortex right here. But in addition to this layer, you also see a layer of cells down here. These are the neural stem cells. These neural stem cells are called radioglial cells historically. They undergo so-called interconnected nuclear migration. The nuclei move up and down and divide at the ventricular surface. And in asymmetric cell division, one of the cells will become neuron. This neuron would actually migrate toward the cortex. Okay? So these cells are not actually born. The cortical neurons are not born in the cortex. They are born in the deep layers, so-called ventricular zone, and migrate out. Okay? So this is the basic of brain development or cortical development. But how do we study this? In 2001, there are several groups in Japan who invented this very cool technique called in utero electroporation. Sounds scary, right? Yes, it's kind of scary. Because we want to understand how brain development, we take pregnant mice, and we can do a C-section to the mother, and then pull out the uterus. And we can inject some material into the ventricle. We can inject DNA, for example, we can inject virus. In this case, we inject DNA, and then zap the brain with electric currents, so that the DNA can be delivered into the cells lining the ventricular surface. And what are these cells I just mentioned? What are the cells deep in the ventricular zone? The neural stem cells, that's right. So the neural stem cells can now get some DNA using this technique. And then we can put the embryos back and then sew the mother up, and everything is fine. The embryos develop normally. The mother can even give birth, okay? So after a few days after this operation, we take out the brain and we look at whether we really successfully deliver these genes. We use green fluorescent protein, or GFP, to test. And this is 2 days after electroporation. We can see a lot of cells expressing this GFP. And these are the cells lining the ventricular surface. And we can also manipulate the gene expression. For example, I was interested in this lissencephaly gene, this one. So So we can use RNAi to knock down the expression of this one and see what happens. Okay, so 2 days after electroporation, and not much difference, right? But if you see 4 days after electroporation, most of the cells have migrated out. Many of them have reached the cortex. However, if we look at the list 1 knockdown neurons, these cells were arrested in the ventricular zone. This is 6 days after electroporation. Most of the cells, control cells, have reached the cortical plate. These cells undergo this one knockdown, still there, deep in the ventricular zone. So it looks like they failed to migrate to the cortex, right? But we have to prove this, okay? So I developed this live imaging of brain slices under a microscope when I was a graduate student. We take out the embryos, section the brain slices, culture these brain slices, and then look at them under a microscope. Of course, you have to have an environmental chamber to control the CO2 level, temperature, humidity, etc. And it took me actually 6 months to develop this, okay? Because cells die, things go wrong and things like that. And then we can take images every 10 minutes, for example, and then put together these images, and it gives you a movie. These neurons migrate in brain slices, and we can see the details. You can see a leading process, and also they put out an axon when they still develop, and clearly we can recapitulate this behavior in vitro. How about when we knock down this one? Clearly you see the leading process continues to grow. However, the neuron, the cell body cannot move. So we prove that if we don't have this one, the neuron cannot move, cannot migrate. So this is quite nice. So we published this actually way back in the 2005, and this actually was my first author publication. So I still like to talk about this because this is where I start. Okay. So we propose a model why neuronal migration is very important. Because if you don't have neuronal migration, the cells don't go to the cortex. Because they don't go to a cortex, the surface doesn't need to expand. So that's why you don't have these convolutions, because the surface of the brain does not need to expand. Okay. And nowadays, dyssencephaly and some related diseases are called neuronal migration disorders because of our research. So it actually have an impact clinically. So this is really, really nice and really exciting. And this paper has been cited for at least 400 times already. And using a similar technique, I actually can resolve the entire pathway. We can look at how these neurostem cells divide. We can even look at the microtubules, the cytoskeleton in these dividing cells. We even use this technique to identify a new progenitor cell. These are called OSVZ radioglial cells. I will talk about this in the afternoon, how I discovered this. So using these techniques, actually, we actually resolve a lot of these behaviors and publish many papers. And this is actually very important because later, in 2016, scientists have discovered that even in the newborn human beings, the neuronal migration still continues. Okay, they get the fetus, they get those children who die from accidents or etc. and get the brain slices, and then they culture them and look at how this neuron migrates. So I think this is actually potentially, we can develop drugs to enhance neuronal migration. If you found the neuronal migration disorders, hopefully we still have some treatments even after birth for these children. So as I mentioned, we published using very similar techniques, resolved a lot of things. we resolved the centrosomal segregation problem in these radioglial cells. This was published in Nature as a cover story, and also we identified these new radioglial cells, neurostem cells, and again published in Nature Neuroscience as a cover story. Okay, but that was all the past. The most difficult part is when you come back to Taiwan. How do you redo this again? Okay, so this is a real challenge because now you are, thousands of miles away from your boss. Okay, your, big boss, big names. How do you do this again? You are on your own. So after I come back to Taiwan, I think I really want to more understand how this brain developed. Okay. But I'm also interested in the clinical side of more brain developmental disorders. So, I think I was lucky. I came back to陽明University at that time. And, our neighbor, Taipei Veteran General Hospital, is a big hospital. And they are very strong in neurology. So I started to collaborate with them. I sit in their seminars and think about clinical applications of my research. And one day, they told me that they are actually looking at a disease called a focal cortical dysplasia. So what does it mean by focal cortical dysplasia? It's called 局灶性大脑皮质发育异常. Why it is called focal? That is because there is only a small part of the brain that was developed abnormally. Okay, the other parts are pretty much normal. So this is actually a little bit different from this encephaly, microcephaly, because the whole brain was abnormal. Okay, so why it is focal? So people have proposed that maybe somatic mutations in these neurostem cells would cause this disease. So we think, oh, maybe this makes sense because you have this neurostem cell, and this neurostem cell will produce a lot of neurons, and if these neurons were abnormal, they will have, very, very severe or very, very abnormal signal sending out to other neurons, and it actually, the end result is that these patients usually have epilepsy, okay? But it is very difficult to find these somatic mutations in the brain, right? So as a basic researcher, I think, how do we move this to a rodent model, okay? And one day I was sleeping, so sleep is very important. I had a dream. I dreamed of a woman, okay? Not Mazu, okay? I'm not going to, come out and elect run for the president. But I dream of this woman, Barbara McClintock. How many of them have heard about Barbara McClintock? Yes, nice. Also a Nobel Prize laureate. She was a really, really pioneer studies with this called transposons. She actually spent most of the time in the cornfield, and she found that these colors of these corns are very unpredictable. It does not obey Mendel's law. So she proposed this transposon hypothesis, and it was not accepted at that time because at that time there's no molecular biology, and the DNA structure is not even known because these are in 30s and 40s of 19th centuries, of 20th centuries. So the DNA structure was solved in 1953, right, by Watson and Crick, yeah? So this hypothesis was not accepted. But after the DNA structure was solved, molecular biology came out, many people have found the same thing in other organisms. So in 1983, she won the Nobel Prize in physiology or medicine. So the concept of this transposon is that there is a piece of DNA that can cut or copy and paste itself and to insert this to another location on the chromosome. This is kind of weird, but it's kind of if you think of it, it's a parasite in your chromosome, right? You just stay there and do nothing, and the only thing it does is jump around. But it actually may help some evolution, right? Because it creates mutations. When it inserts into some places, it may actually translocate genes, it may interrupt genes, and that's why these color of the corns are, going crazy. And nowadays, transposon has been used to mutagenize genome of many organisms, including Drosophila. And I think, yes, can I use this concept into a mammal? And we all know that mammals are way more expensive than Drosophila, right? A cage, only 5 mice, and you need to spend a lot of money. So it is very difficult, very expensive to do this transposon screening. But I had this idea. I have learned in neutral electroporation, I have transposon. Boom. Can I electroporate these transposons into neural stem cells? And then allow the neural stem cell to develop. And if the transposon hit an important gene in this process, then something will go wrong. And remember, these neurons have to go to the cortex to have their functions. So, I woke up 4 o'clock in the morning and I run to my computer, type in transposon, electroporation, brain development, in PubMed, and it came out 0. I've done research for many years, and every time I have some idea, and I'm sure many of you have the same experience, and I go to a computer and PubMed the keywords, and it comes hundreds or thousands of hits, right? It's already been done. Okay, but this time 0. So this is really, really exciting. So I emailed my lab members, and we will have a lab meeting 9 o'clock in the morning. Okay. I told my students with this idea, and one of my graduate students, PhD student, took this challenge. Okay. So she electroporated these transposon into the brain, and look how these neurons behave. So in the control, as you can see, most of the neurons will migrate to the cortex already when we electroporate this transposon alone without the enzyme. It still requires an enzyme. These neurons still can migrate. But if we electroporate both transposon and this enzyme, then you see an additional layer underneath. So we are doing a genetic screen on a cellular level in a single mouse. Okay? So this is really, really exciting. So we can go through some molecular biology. This has been established. We can actually take out this piece of tissue and then go through sprinkler PCR to amplify the neighboring piece of DNA, and then we know exactly where these transposons interrupt. So using this technology, we identified 33 new genes that might be involved in this process. Of course, some of the genes have already been implicated in human development brain developmental disorders, which is actually exciting because that means that our method worked. Okay. And of course, then we throw this, and actually recently, one of the genes that I presented this in International Epilepsy Congress, and one clinician came up to me, so telling me that they identify a patient with mutation in our gene list, which is really, really exciting. So we are now collaborating with the Israel group to study the function of this gene. Very exciting. So then we also throw this this gene list into a computer and do some analysis and to identify what the most function these genes are involved. These are mostly involve development. You see, these are really development genes that even give us more confidence. But we still need to verify whether these genes would really cause brain developmental defects in brain development. So we, again, electroporate many shRNAs for many of these genes. And you see, indeed, many of these genes, when we knock down, you see a blockage of neuronal migration. We can rescue this using cDNA again. And this is really hard work because we have 33 genes to test, and we did about 25 of them. And you can see that these are the cells arrested in the deep layers, and you can see normal neurons have mostly migrate. 2/3 of the genes, when we knock them down, you see an accumulation of cells in the deep layers. That gives us more confidence that these genes are involved in brain development. How about our patients? So we go back to clinicians, and since this idea came from FCD, so we need to go back to the FCD patients. So we were able to access these tissues because these patients suffer from epilepsy, and one of the treatment that the clinicians can do is to take out this piece of tissue because these pieces of tissue have some problem. Okay, so they use MRI, EEG, PET scan to localize this piece, and then actually they dissect out this piece of brain, and then the epilepsy is gone. Okay, so we then have the privilege to collect this piece of tissue and can do some genetic analysis using next generation sequencing. We do a deep sequence because these somatic mutations may be very rare, hidden deep in the normal neurons. So we had to go through a pipeline of these next generation sequencing and identify those rare variations in the brain tissue. And at that time, we only had money to do 6 patients. And among these 6 patients, we actually identified at least 3 genes that overlapped with the genes that we found in the rodents. So we, again, knocked them down and see that indeed those gene knockdowns were actually causing the neuronal migration defects. We can, again, rescue this phenotype. And one of the patients, it has a somatic mutation, a missense mutation. When we deliver this missense mutation, the cells, this mutation cannot rescue these migrations. And indeed, telling us that this mutation really did cause the focal lesion. So this was actually published 3 years ago now by my talented postdoc, PhD student Yiling, and also many other students, both doctoral and master students. And now, actually, she and her husband, Joshua, are now postdocs at UCSF. So very outstanding. Okay, so these are the genetic screening. And now I want to switch gear a little bit to talk about the mechanisms. So, we talk about, okay, this gene, that gene. How about mechanism? How about basic research, which can, help understanding the molecular mechanisms of these brain developmental disorders? So I will start talking about my favorite gene, this one. So why mutations in this one would cause lisencephaly? And we already know because migration was blocked. And then you can step, one step back and think, then why this mutation will cause neuronal migration defects, right? So that's the, very basic question that any graduate students can ask. So then I have to go back to the literature and understand the mechanism or the known mechanism of neural migration. And when I look at this one, And I found that actually a lot of people have been studying the molecular function of this one. It interacts with thymine, the motor protein. The motor protein would interact with microtubules. The microtubule is centered at the centrosome. So I think, maybe the centrosome is kind of important. So I, look up the literature. What is the relationship between the centrosome and neuronal migration? I found these few papers talking about the centrosome needs to migrate into the leading process and the nucleus will follow. Okay. So this is very, very interesting during brain development. So I think, maybe I should look at how centrosome move in our lissencephaly, deficient neurons. So then I used my own trick, my brain slice culture, and then triply label these neurons with 3 different constructs. One is the cell body, and another, the centrosome, and then the nucleus. I need to reproduce the literature, right? So, they use in vitro model. Now I use brain slice model. So using live cell imaging, you can see how this centrosome move into the leading process, and the nucleus follows. Amazing. You can see it under your microscope, and I can watch this for hours. This is so interesting. Okay, so I'm going to talk about how excited I was when I first saw this, because this is really what have driven me, to today's status. So this is really exciting. Okay, this is even more exciting than publish, then your paper get accepted by a prominent journal, because this is the essence of the excitement. Okay, so you are the first human being to see this. How exciting! Very exciting. So we can look at the centrosome, how this centrosome moves using so-called chymograph. The centrosome moves smoothly. The nucleus would move in a saltatory manner. It stops, moves, stops, moves, and stops. There are still many, many questions. Why does it stop? Why does it not continue? Something like that. How about when we knock out this one? Okay? So the centrosome is here. If you can read the chymograph, you already know the answer. The centrosome cannot move. Wow, so that's why the neuron cannot move, because the centrosome cannot move. Okay? So then I did a lot of other assays to show that the centrosome, the thymine, this one, need to be localized to the leading process and then pull the centrosome forwards, but it's already published, so you can read this online. So since the centrosome is really interesting, so I'm going to talk about more my recent work. This is also by accident. So we have been collaborating with physicians who have a cohort of epilepsy patients, neurodevelopmental patients, and lissencephaly patients. So usually we did some clinical, some genetic counseling, and do a whole exome sequencing or a panel or a single sequencing to tell them, what mutation does this children have. But occasionally, we could not find known genes, we could not find answers. So we had to use whole genome or whole exome sequencing to identify potential mutations. And this is a really, really interesting case, because we found a Taiwanese patient with this CEP85L gene. I've never heard about this, okay? I never heard about this gene, but it was suggested by its name. It may be a centrosome gene. So I was, okay, this might be interesting, because I studied centrosome before. And since there is only one case, we were still not sure. So we then call out and to other collaborators, other centers with these patients, and tell them that we found this, and did you also find this similar phenotype with the same variants? It turns out there are dozens of them, okay? And this patient would have so-called posterior dominant lissencephaly, the posterior part is more severe, the frontal part is a little bit less severe. And these patients can be inherited or de novo, or maybe this might be a germline mutation. And interestingly, among the patients we have, the variants all occur in the N-terminus of this gene. And we still don't understand why this is the case. We're still investigating this. So apparently this domain has important function on the brain development. But the first thing first, we need to show that how does mutations or variants or defects of SEP85L will cause this encephaly. And the first thing we tested was, of course, neuronal migration. So we knock down SEP85L and look at how these neurons migrate. And not surprisingly, we found that the cells are still arrested at the white matter. In control cells, you see most of the cells are already in the cortex. We also look at the differentiation by staining these neurons with neuronal marker nu-n. We saw that at this stage, about 40 to 50% of the cells have expressed nu-n, which looks like yellow. In those arrested neurons, there are still about the same number of neurons expressing nu-n. So the differentiation part is not much affected. And then we want to go into cells, just like what imaging usually do. See how these cells express this gene and where does the protein localize to. So, of course, as suggested by its name, we firstly look at the centrosome. So we deliver this gene into the culture cells, and then also staying with centrosomal marker, we found that the red signal is the SEP85L. It's very close to the centrosome, very close to the centrine, but not 100% colocalized. And we also use other centrosomal markers, centriolar markers, and we actually found that the SEP85L is actually localized at the so-called proximal ends of these centrioles. Okay, this is done by Dr. Wang Wanqing, a very close collaborator at MICU. And also this is true in different cell cycle phase, for example, G1 phase and S phase. And also we stain the brain slices, and we found that the SEP85L is really localized to the centrosome. So it does, it is a centrosomal protein. So this is actually very, very cool. And we also use the 3D structure illumination microscopy, or 3D STEAM, the super resolution microscope, and look at the localization of this SEP85L in the centrosome. And as you can see here, it looks like a ring at the proximal end of the centriole. So this, is unpublished and really, really exciting. We will then go into the details. What's the function of SEP85L in maybe centriolar genesis or things like that. So, but we have to publish quickly. So we published the first part of the story about the genetics and also the animal studies in neuron just last year. Okay, so this is really, really exciting because we have many patients and this gene seems like very important. Okay, then I'm going to talk about another gene, okay? This gene is also very, very different. It's not a centrosome gene. And it is actually a surprise to us. So again, in a Taiwanese patient with lissencephaly, and this patient actually has not only lissencephaly, but also muscle weakness, the lower limb weakness. So also, there is no known gene variation in this patient. So we did a whole exome sequencing again to identify potential variations. And to our surprise, we identified a mutation in this BICD2 gene, and this is actually a Drosophila homologue of bicaudal, a very important gene for development in Drosophila. But it puzzled us because BICD2 mutations does not cause brain developmental disorders. It actually causes another disease called spinal muscular atrophy, a particular type, so-called SMA-LED2. So this is strange because although it has a little bit of muscle weakness, but none of these patients have brain malformations. So we then look at what are the other mutations in these SMA patients. It turns out these patients all carry so-called missense mutations. These mutations will only make an amino acid residue change. But our mutation is a truncated form mutation. So this one is truncated at the position 775. Okay? This may be why this mutation is causing more severe phenotype. But again, I'm a basic researcher. So this is when we come in and then tell them whether this hypothesis is true. So we then express the mutant forms of BICD2 in the rodents. Again, we use in vitro electroporation and electroporate these variants. When we express this wild-type BICD2, it does not affect neuronal migration, of course. When we have this truncated form, you see super severe neuronal migration defects. If we have this SMA-associated mutations, the neuronal migration are normal. Okay, that's the first step. And then we want to know why this migration is defective. So again, we look at the centrosome, although it's not the centrosomal protein, but we are really interested in knowing what's BICD2 is doing inside migrating neurons. So as you can see here, in normal cell, the centrosome migrate, the nucleus migrate, no problem. Here, when we express this truncated BICD2, the centrosome have moved to the leading process without problem. So it is different from this one mutations. But the nucleus cannot move. So the mechanism is definitely different, although it is, they are all both involved in neuronal migration. So what is BICD2? BICD2, it turns out, is again a dynein-associated protein. Not all the time, but sometimes it binds to dynein. And in certain cell cycle phase, BICD2 would actually bring dynein onto the nuclear membrane. So when you express BICD2 and then it's localized to the nuclear membrane, also when we look at dynein, it's on the nuclear membrane very clearly. When we express truncated BICD2, you see a lot of BICD2 were dispersed in the cytosol. Also, dynein is dispersed. When we express SMA-associated BICD2 mutants, you see they did not affect nuclear membrane localization. Okay? So we can also prove this by using fractionation. You have a cytoplasmic extraction, nuclear extraction. You see, dynein heavy chain, in the mutant is less, okay, than the control, and also DIC less. BICD2 truncated form does not localize to the nuclear membrane. So what is it doing? So it turns out that we showed that BICD2 would interact with dynein complex, and it interacts with another protein called nesperin-2. It interacts with this nesperin-2. And nesperin-2 is a nuclear membrane protein. So BICD2 is responsible for bringing dynein onto the nuclear membrane. And when the dynein is localized to the nuclear membrane, it actually pulls the nucleus along the microtubules and make the nucleus translocate. When we have a truncated form, it binds to the nuclear dynein, but it does not bring dynein onto the nuclear membrane, so the nucleus cannot move. Okay, so we can do a lot of things using our animal model. So in the first part of my talk, I really want to emphasize how basic research can solve clinical problems. Here we found that encephaly is caused by neuronal migration defects. And why mutations in certain genes will cause neuronal migration defects? Because they are involved in different steps during neuronal migration. For example, dynein LIS1 is localized to the eating process, and then it pulls the microtubules. And because the microtubules are centered at the centrosome, so the centrosome can move. We still don't know what's the function of CEP85L in this process, so we still have work to do. And the second step is dynein LIS1 and BICD2 needs to bind to the nuclear membrane and then translocate the nucleus to catch up with the centrosome. And of course, other monoproteins, for example, myosin 2 is also involved in this process. Okay? So can you imagine over the past 10, 15 years, we have solved a lot of problems like this, okay? By using our very advanced approaches. How much time do I have? 10 minutes? Okay. The second part of my talk, I will talk about, again, use optical imaging. Can we do more? And now we turn our focus to the cerebellum. Okay? Now we have cerebral cortex and cerebellum. And we focus on this medulloblastoma, which is also a very important clinical problem because it's a very common tumor in the brain in children. Okay, one of the commonest. In one type of the medulloblastoma called Sony hedgehog, actually Eugene is very, very familiar with this. It actually has this overexpression of a transcription factor called ATO1. But people do not understand why ATO1 overexpression will cause this medulloblastoma. So a French group who is studying the cell biology of medulloblastoma visited Taiwan in 2013. And incidentally, we meet with each other and presented our work. And I told him that I had this electroporation and migration thing. And he said, can you do this in cerebellum? I think, I've heard about this work in Japan too. I think people have done this. So I went to Japan and learned this technique, so-called cerebellar electroporation. And you can actually do this in the cerebellum. Again, these neurons, progenitor cells, and these are the migrating granular neurons. Okay? And we can use this technique to label progenitor cells and how these progenitor cells differentiate and migrate to produce granular neurons and using this electroporation. And of course, this process has been known for at least 100 years. But can we do better? Of course. We can express genes in your progenitor cells, right? And remember, I told you that ATO1 is overexpressed in medulloblastoma. So we try to overexpress this gene in these granular neuron progenitor cells to see what happens. Can they become these tumors? So as you can see here, these are the postnatal day 6, and then it's day 1, day 2, day 3, day 4 after electroporation. You can see cells migrating inwards. Okay, but when we overexpress ATO1, you see a lot of cells are still arrested it turns out they keep proliferating in the external granular layer. And what is the mechanism? Because it is Sony Hedgehog type, and if you heard about Sony Hedgehog, you know Sony Hedgehog signaling has to go through primary cilia. So we decided to look at primary cilia. I think it's Eugene's favorite organelle in the cell, right? And we found that actually these primary cilia are highly localized at the external granular layer and associated with these neuroprogenitor cells. After they differentiate, you can see these red thing, these little sticks, will start to disappear. Sorry, the yellow things will start to disappear. So it is very interesting that these progenitor cells have primary cilia, and then when they differentiate, they have to retract primary cilia. Okay, so primary cilia has a lot of functions in transducing many signals, mechanical signals, Sony Hedgehog, etc. So we think that since it is highly associated with, progenitor cells and ATOL1, does ATOL1 have any function with the ciliogenesis or retraction of this primary cilia? So that we knock down ATOL1 and overexpress ATOL1. We found that when we knock down ATOL1, the cell, the primary cilia were retracted. When we overexpress them, then there is more primary cilia. So we then work out the entire molecular pathway. I don't want to go into the details, but the conclusion is that when you have ATOL1 expression, it actually will help grow primary cilia in progenitor cells. And the Purkinje cells would express Sony Hedgehog and release the Sony Hedgehog signaling and keep the progenitor proliferating. When they decide to differentiate, it retracts the primary cilia and then differentiate and then migrate to the cortex, internal granular layer. When you have an overexpression, a continuous expression of ATOL1, the primary cilia cannot retract, so that it actually keeps sensing Sony Hedgehog signaling, and then it keeps proliferating. And when a neuroprogenitor cell keeps proliferating, what do they become? They become tumors, right? So this is why ATOL1 is causing medulloblastoma, because of the primary cilia. Okay? So this is another paper, collaborative paper with a French group. Okay, so 2 minutes. I'm going to talk about my physics background. So as a physicist, I also had this really cool technique, so-called two-photon microscope. And it actually can use this two-photon microscope and look at these neurons underneath the skull. We can do a cranial window, okay, or thin the skull, and then use two-photon microscope. I'm sure Dr. Johns also used this technique to look at his neurons before. And underneath the cranial window or the skull, you can see these neurons. And actually, these neurons actually electroporated in utero. Okay, so after birth, the neurons are still there. Okay, and these are in live animals, which is really nice because we can take the mouse out and look at the neurons and then put them back, and then 2 days later, 1 month later, you still can identify the same neuron, same dendrite, even same dendritic spine. Okay? But it is very, laborious, labor intensity work. So we started to develop this AI assisted labeling of these so-called dendritic spines or these synapses. And this is a collaboration with Huashou. Okay, and hopefully next year we will launch this and do a service for people who are interested in identifying these dendritic spines using our AI model. And as a microscopist, I'm also really fascinated by all the live microscopy. This is a microglia underneath the thin skull prep in a live animal, mouse. A mouse just lying there. Okay? And you can look at how these neurons or other cells behave in the brain. And again, this is very important for neuroinflammation, neurodegeneration, and things like that. And we collaborated with a startup company called Elixiron, developing drug targets to the microglia, and we are hoping to use this drug to inhibit microglia inflammation in Alzheimer's disease and other neurodegenerative models. In Alzheimer's disease, using this drug, you can see a decrease in the plaque formation, okay, which is really exciting. And we also developed algorithms to analyze these microglia pathology. So, I myself is also a programmer, so I can actually write computer code. Okay, I think that's the end of my talk, and we published papers in high-profile journals, including neuron, developmental cell, nature communications, etc. And we also welcome collaborations because especially clinicians who are really interested in understanding the mechanism of diseases, and for basic researchers who does cell biology and biochemistry, but we have animal models. If you want to verify this in animal models, we still can help. Okay, I'd like to thank all my collaborators and all the sponsors and also the companies that support this work. With that, I'm happy to take questions. Thank you very much. Thank you.
Thank you, Professor Tsai, for his wonderful talk. We'll first start from students. Does anyone have any question?
Give hearts.
Yes, that one. Okay.
If you feel more comfortable in Mandarin,
yeah.
still you can ask questions in Mandarin.
Okay, thank you, Professor. Very exciting talk. Thank you. I have two questions. The first question is, I'm wondering, what is the driving force to guiding the centrosomes to go to the specific position? Is that already be the niche is controlled by the genes or another chemical reactions?
This is an excellent question. Yes, we have a model, but we don't have a proof yet. So here, you have a so-called swelling, okay? So I did not talk about this. The swelling will form right here, okay? We found that the dyne in this one is not in the tip of the living process. It's very specifically localized in the swelling, okay? But why dyne is localized here? We don't know. We think that maybe there are adhesion molecules concentrated here. And the adhesion molecules will recruit dyne to this place. Okay. So why do the neurons have formed a swelling here? We also don't know, but we think that maybe because when it migrates, there are actually radioglial cells. It needs to attach to the radioglial fibers, okay? And when it attaches, it will gradually form a kind of adhesion site. It's not focal adhesion, but it's a kind of adhesion site here, okay? And then it recruits dyne to this position, okay? And dyne does this, pulls the microtubules up to the healing process. And you have a second question? No? You have surrendered microphone.
if this kind of niche is already being controlled by genes or another factor?
We don't know. It may be random. I think it's probably random. Okay, just like when you do rock climbing, right? You see whichever place is more easy to grab, you will grab on it. Yeah. So I think there may be ways to study this, but because I think this distance is not constant. This distance may be random. Sometimes it's a little bit further. Sometimes it's a little bit closer. Yeah. So maybe it's a stochastic event.
Thank you, Professor.
前几个比较难学，你们是要给 hip hop 的关系。
Thanks for your speech. You say knock down this one inhibits neural migration. And how about overexpress this one? If this one overexpression, neuron cell would develop more quickly or the same as normal?
Excellent question. So the answer is if you have too much this one, neuronal migration is still inhibited. And it actually, if you have too much, then we see the cells are cannot migrate and actually will lead to cell death. And there is a clinical side of it because the Israel group identified a triplet, this one gene. So this person would have 3 locus. And this patient actually have a microcephaly plus lissencephaly. So you cannot have too much this one. Okay? So too much, too less, no good.
Thank you.
Thanks for this amazing speak. And I'm very interested in the relationship between the transposome and neural migration. And so I would like to ask, how do Professor find those 32 transposome genes, and why did you choose 25 of them to do this kind of experience? And does there have a similar function between these genes, or they have a different function? Thank you.
Okay, so these genes were selected, not selected. Actually, these are the normal genes that CAPS-A, PM2, SEMA3, 60, things like that, and MACF1. These are done by using this sprinkler PCR. So the transposon will jump randomly, okay? Then you insert into your genome, okay, and sit there, okay? And then we amplify the neighboring sequence and neighboring segments, and then send it out for sequencing. And if it is localized at the gene region, then we call it, okay, this gene is affected. Okay, so this is how we select out these potential genes for neurodevelopment. So these are not so-called transposon genes. These are actually genes identified by transposon. Okay. And do they have similar functions? They have centrosome thing. They have, I think some signaling or some, there are also kinases, Golgi, adhesion, things like that. But they very often is involved in development. And is, for example, less involved in, for example, cardiovascular, connective tissue, cancer, and things like that. Okay, so they are not doing only one thing, but as you can imagine, neuronal migration requires signaling, requires surface receptors, requires cytoskeleton, and when you modify cytoskeleton, you need kinases, centrosome skeleton, and things like that. So you will get a diverse genetic functions, but they may be all involved in either differentiation, migration, or things like that. Okay, so this is the power of 4 genetics, as Dr. Johnson would agree that. Using Drosophila, I have identified so many important genes in development because of 4 genetics. I don't know how to select, so you are the boss. Yeah, the one with the microphone.
So thanks for a wonderful talk. So we know that family history plays an important role in many brain disorders. Is it possible for us to screen the disease in some vulnerable embryo and during the brain development and induce some protective mechanism through the in utero electroporation?
Yes, these are very, very often asked questions. Yes and no. Of course, we can all do genetic screening, okay? But these are all rare diseases. This only happens, 1 in, 80,000 or 100,000. Okay, so quite kind of rare. And you can screen everyone, but how much you are going to pay? Who is going to pay for this? So this is the question. Okay, it's not a scientific question. Yes, we can, but economically, is it worthwhile? Okay, and it can be inherited sometimes because another gene called BCX double 14 female can be carriers. The symptom for the female is less severe. Okay, but when the boys carry this BCX mutant, it gets disencephaly. So that can be done because if you have one boy disencephaly patient, then you will think, okay, can we have another children? Okay, so that can be done. That's more efficient. But for this one, for disencephaly, this one is mostly sporadic. Okay, why is this sporadic? Because you have the disease, then usually the patient die in their teenage. They don't get married, they don't have siblings, they don't have offspring. So usually these are the sporadic de novo mutations. Okay. So it's even harder to screen. Yeah, because the parents are normal. Okay.
Can we induce the protect mechanism through the in utero electroporation?
Yeah, that's a dream that we have. But also, again, how do you identify the patient? Because the gyri develop very late in development. Okay, so it's usually 20, 30 weeks after after pregnancy, like 26 to 30 weeks. It requires very, very experienced radiologists to use ultrasound to know that, this baby may have some problem with their gyro formation. Yeah. So yeah, we're hoping that we can do something to this patient at this stage, but still so far not much has been done.
This will be the last one. 1 second.
With me talk.
But you are welcome to ask more questions.
Don't tell them.
Well, thanks for the talk. I've been studying for the neuron regeneration and the axon extension. So I'm wondering whether this kind of neuron migrating defects will affect the effect of the process during the axon extension to the matrix area.
Yes, they will. I'm sorry I didn't have enough time to talk about this, but imagine, there is the axon right here, and actually the axon here will become much shorter, and dynein, this one, is also involved in this process. So if you go back to my 2005 JCB paper, it talks about the defects in axonal growth, and also we had a review paper saying how this one dynein will affect axonal guidance. Okay.
Maybe we can take some questions from the faculty members and then back to students if there are more questions.
Yes, please.
Hello, Jim. Thanks for your wonderful talk. I'm just curious about the neuronal cell migration. So why centrosomes and nucleus they need to migrate in kind of stepwise manners? Like a centrosome move first, then they stop, and for the nucleus, then centrosome just have a chance to move again. Why they just they move together will be more efficient, right?
I know. I don't know. I mean, I think only he knows. Yeah, I think there might not be intentional. Okay, maybe that's my just out of my brain. Maybe it's the matter of because resistance. Because sometimes we do see the centrosome and nucleus move together, but most of the time, they move like this. I think maybe it's a resistance because they don't move in the vacuum. They move in a very densely packed tissue, right? So the tissue, there are so many other cells around them. So when you pull, you don't have enough force, right? Because you have only a few, maybe a few hundred dying over here, and when you try to pull everything, and then you get stuck. Well, I cannot go, right? So that's just a guess.
So it's possible to test this hypothesis in the
think
condensed
so.
matrix or soft
Exactly.
matrix?
That's actually when I was a graduate student, I wanted to do this. You can change the density of the environment, and then to test whether this is really true. And yeah.
In the development of the brain, which part of the brain is first developed in terms of structure and function? Can you tell is the basal ganglia, or is the brainstem, or which part, or hippocampus or something? Can you identify which part of the brain is the first developed?
I think so, but it's a thing that that's a thing that I need to go back to the textbook and read. I don't have a good picture here, but actually the cerebral cortex developed very, very late, okay? And the brainstem and the hippocampus, even during evolution, okay, they appear first. For example, turtles, reptiles, birds, they don't have this so-called cerebral cortex. Only mammals have cerebral cortex. So it appears very late in the evolution. Yes. So I think there is a, I don't know how this gene, maybe some mutation in these different evolution genes that will cause the development of this frontal part, this telencephalon. And it's actually a big question in this field. Yeah. So the brainstem and all that are developed very, very early, and the cerebral cortex developed very late. Yeah. So, but I only focus on this part, for many years. Yeah. Okay.
Question is, which part of the brain that the by-CD mutant is affected to cause only muscular atrophy? It's not
Yes.
brain function.
Well, yeah. It actually has a dominant effect. So it's a hemizygous, or not, the heterozygote will cause this. So I don't know what kind, it may be gain some functions in binding some unwanted protein in the muscle, but I did not look deeply into that mechanism. But I think it's been studied before. So it may have some kind of gain of function effects on this protein.
Thank you for a nice talk. So I'm wondering about the effect of gene that you found that will affect the migration of the neurons. So are those genes like neuron specific or expressed in a particular developmental stage? And a follow-up question regarding this question is that if they are not specific in neurons, and do they affect, or when there's a mutation, do they affect like glia cells, any type of glia, and does that affect the neural development?
Excellent question. So many of these genes were not specific to neurons. Okay, for example, this one is ubiquitously expressed in the whole body. All the cells have this one expression. But why? Because the defects in the brain more severely, we don't know. But as we remember, this one mutation, okay, this patient they are heterozygotes. Okay, so these are called haploinsufficiency. These patients only have 50% of this one. But that's enough for survival. Okay, most of the cells survive okay with only 50% of this one. If you have total knockout, embryonic lesion, like die very early in the mouse, probably die in the what is that called? Even before the brain form. Okay. So it's a very important gene, but if you have one copy, that's good enough. Okay. So why it affects the brain, so much? Maybe, okay, this is again a guess. Maybe neural migration is so important that if you have any defects, it does not prohibit the formation of the cortex, right? It's still there. So if you see it from 500 meters away, you see this person still have a brain, right? So it's not affecting too much. But because the human beings are so intelligent, it requires the proper migration. Okay, if you have a mouse, the mouse, 50% of this one, the heterozygotes, the phenotype very, very subtle. If you do some water maze, they are still okay. Okay? You only have to challenge them with mathematics, before you find any phenotype. Okay. So this is why the human being is very fragile, very, very, very sensitive to small mutations, because we are so intelligent, we are so, highly refined. The second part is that because neuronal migration cannot be catched up, why? These radioglial cells will disappear after birth. They become astrocytes. Okay? So, when they don't have this guidance, they don't know where to go. Even they try to catch up, but the guidance is no longer there. So they just stop there. So it looks like this. So you have a very, very thick cortex, right? So maybe it affects all cells, but because neural migration is so important and so needs a very, very timely control, so it affects the brain most severely. Okay. And molecularly, why this one is important? This one is not involved in small vesicle movements. So when Diney needs to walk in or have a small lysosome mRNA, when it carries these, it's okay without this one. But if you have a heavy load, for example, pulling the centrosome, very, very heavy because you are pulling the entire cytoskeleton. Pulling the nucleus, very heavy. Then, with addition of this one, it actually increased the power stroke and also the efficiency. So that has been published by my advisor in Cell and other journals using biophysics to answer these questions. Okay. Other students? Come on.
Are there any more questions? I saw some hands, yeah.
Yes. That's,
Thank you for your speech. And as Professor said, the transposon can give a scan of the target gene related to neuron migration. I'm curious about that, whether the genes such as CEP85 and BICD2 are detected in your transposon results.
excellent question. We did not see that in our list because we only use a very limited budget and limited mice, and I identified a limited number of genes. And we just, the first paper we want to show was this is doable. Okay, proof of principle. Okay, it has not been saturated yet. What do I mean by saturated? Because if you do enough number of mice, then you will find, you start to see certain genes over and over again. You don't find any more new genes. But it would be very expensive in mice to do this. In Drosophila, yes, you can do that saturation. But in mice, so that's why this paper is not in Nature Neuroscience or Nature, because they criticize us. You don't have enough mice, you don't have enough patients. What can we do? We don't have enough budget.
So NGS can replace the process that transposon.
Yeah, so we do it in two ways. One way is from animals, the other way from patients. But remember, from patients, you do a whole next generation sequencing. You get hundreds and thousands of potential hits. You don't know which one is meaningful. And our 33 genes give you some guidance. Okay, look at these 33 genes. Maybe you get your hit right here. Yeah.
Thank you.
Okay, thank you for your presentation. So in your presentation, we see a lot of development diseases associated with neuron migration and all the troubles in the neuron migrations. And I'm wondering that after we would become like teenager and adult, our brain is still changing. So my question is that does this neuron migration still happen in like teenager brain or adult brain, or does this has some implication in like neuron damage and maybe the neuron just move to the damaged part and fixes? So this is my question.
Yeah, that's excellent question. Honestly, I'm not an expert in adult neural migration or injuries. But this paper, they show that neural migration still occurs even 6 years after birth in human. And I don't know if they are doing even older brain slices. Okay, so this paper is encouraging us because neural migration still happens, for a certain period of time after birth. But I don't know whether in teenage or in adults do these neurons. What I mean is the cortical neurons. Do they still migrate? Okay. In my experience in mouse, they don't move too much. Because using our two, cranial window, I still can see the same dendrite, same spine, same neuron at the same place. If they migrate, I would detect, So maybe there are migration, but not so much. But in the mouse, at least I know, there is a very, very interesting pathway called rostral migratory stream, RMS. That is very special neural migration. That is the neurons they will generate from subventricular zone, SVZ, to populate neurons in the olfactory bulb, and they have to migrate all the way to the olfactory bulb to replenish some of the dying neurons in the olfactory bulb. And I don't remember whether human beings also have this mechanism. So in some particular cases, neurons still migrate in the normal. Yes.
Thank you for your wonderful talk. I have one question. You have identified 32 genes related to brain development. However, sometimes double mutation in different genes cause disease instead of one single gene. So I wonder whether some important genes are ignored in your transposon approach.
Yes, very, very good question. Yes, we don't know whether some of the genes can cause neural migration defects, but we did not detect them because a single mutation or single dysfunction may not affect enough. Okay, and also using our technique, because we mix up all of the cells, so it's possible for us to know whether this transposon or this mutation occurred in one cell or in different cells. Okay, so that's why we did this verification one by one. Okay, and for those we can do 2 by 2, but you have to do combination. And my student refused doing this experiment. Because she wants to break it sooner than later. Yeah, but yes, good question. Some of the mutation may have to occur, simultaneously to have the effect. So we haven't done it, and we are busy enough, right, with these genes. Yes.
Thank you for your inspiring speech, and I would like to ask a question. And it's that during the process that the nucleus migrates, if the two cells fuse together, what will happen?
The two cells fuse together. Interesting. I never thought about this. I don't know. I don't really know if the two cells fuse together, how the nuclei would migrate, or relative to each other. Yeah, but interesting question.
Any other questions? No. When that student asked about the
Thank you for the interesting talk. And I have a question which may not be directly related to your talk, but I'd like to ask if rodent models are available for studying the functions or development of neocortex.
For the what?
Neocortex.
The rodent model is available for
studying the function or development of neocortex.
Neocortex. Yeah, of course. The rodents, this part is called neocortex. Yeah. Well, everything we talk about is the neocortex. Yeah. Everything we talk about. So this part is a neocortex. Okay. So we study this part. Yeah. So this is where our. I hope this actually this part, right? It's called neocortex. We electroporate the most efficient part is when we can electroporate into this part. Okay. And I know that, and there are now people who are like studying the peripheral cortex down here, but yes, this is the interest of area that we're studying.
Okay, if there are no more questions, that's all. Thank Professor Cai.
Thank you. It's my great honor to be here.
下午的那个学思历程是在那个生科导读的那个教室，然后如果有同学有兴趣的话，也可以就是去教室里面，只要有位置都可以坐下来，就可以问一些跟那个就是 生涯比较相关的问题，比较我的学术。